Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about these principles of selective sweeps and hitchhiking. What happens is, if you have a region of low recombination in particular, when an advantageous variant spreads through the population, it eliminates variation at nearby neutral sites. It reduces pi, which is our measure of nucleotide variation. Now, what I'd like to do in this video is explore how we can see signatures of past natural selection in the genome leveraging this. Now again, recombination can help us detect natural selection. So we're going to use the principles we talked about in the last video to actually identify areas that seem to have experienced natural selection. Now an important principle which I haven't emphasized up until now is that recombination rates are not constant across the genome. There are areas where recombination is very common, and we already discussed in this context crossover hotspots, but there are often broad regions which have many hotspots, and there are broad regions that have almost no hotspots. So recombination is especially often thought to be low near the centromeres. The centromere is the part of the chromosome that holds together the chromatids. So let's imagine that here's a chromosome, that you have your centromere here at one end, and let's say we're looking at different stretches of a million base pairs. So this first stretch of a million base pairs, let's say the recombination rate is 0.1 centimorgans. So that's a very low recombination rate. We go a little bit further out in this stretch of a million base pairs, the recombination rate is 3 centimorgans. Go a little bit further out in this stretch of a million base pairs, the recombination rate is 5 centimorgans. So an important principle I want to stress from this is that you can't directly translate centimorgans to millions of bases or numbers of bases because there's not a one-to-one transition here. There are areas where very low centimorgans are associated with high numbers of bases. There are areas where the number of centimorgans is much higher for the same number of bases. Okay? The linear order stays the same either way, but the recombination rate may change. And again, as I mentioned, the recombination rate is often lower near the centromere. Now we measure recombination rate as centimorgans per megabase. So let me show you an actual picture of human recombination rates. This is looking across chromosome 3 of humans. So you see here near the centromere, recombination rate is low, but here on the x-axis is position in millions of bases. On the y-axis is the average recombination rate. We see here near the end, the recombination is pretty high, goes down, down, etc., but low again near the centromere. So it's very variable. Now what we can do is we can focus at what's going to happen if you have an adaptive variant in, say, a region like this, where recombination rate is very low, versus if you have an adaptive variant in a region like this, where recombination rate is moderately high. So let's look at that. Again, these new adaptive alleles can arise by mutation every now and then, and sometimes they arise in a region where recombination is low. So again, there's low centimorgans per megabase, no hotspots. So in this case, now we have 5 variable sites depicted here on this figure. They're in blue. I apologize if it's a little bit hard to see on the slide, but I wanted to show you an actual stretch of sequence there. So let's see what happens. Let's put this now in the case where there's no recombination. So here's our new adaptive variant. This is this beneficial G. If there's no recombination, what's going to happen? Well, these stars indicate the variable sites. So there's 1, 2, 3, 4, 5 different variable sites aside from the selected ones. So these are ones we're assuming are neutral, that it does not matter for fitness if you have a T or a G here, or if you have a C or a T there. So as this G spreads, you lose variation at all other nucleotide sites. So now none of these are variable anymore. That's why I got rid of the stars. So you lost all variation across this entire stretch. Pi across this whole sequence now, or across this whole stretch, is 0 in this population. Now let's try this again in a region where recombination is very common, where you might have high centimorgans per megabase. Well, here's that adaptive variant again. We still have 5 other variable sites with neutral variation. Well, as it spreads in this case, you see we've already in this case had some recombination out near the ends, right? Well, as it continues to spread, we've lost variation very close to the selected site, right? That maybe there was no recombination between these two, but on average, since there's a lot of recombination, you retained variation out here, you retained it here, here, and here. So again, you maintain a high pi across most of the sequence, but you lost a tiny bit of pi because sequence is very close to the spreading site lost variation. So we can generate a prediction. Our prediction is if you survey regions around the genome, then you should be able to find some sort of association between recombination rate and pi. So take this quiz question real quick. Do you think this relationship will be absent? Do you think there's really no relationship? Do you think regions of high recombination should generally have high pi? This would be a positive association. Or do you expect a negative relationship? The regions of high recombination should have low pi. Well, that was an extremely easy one. Again, as I said, if you have high recombination, you're able to sustain variation. So we do expect this to be a positive association. And in fact, that's exactly what you see. So this is looking at data from humans. On the x-axis here is recombination rate. On the y-axis here is pi. This is from a study by Hellman et al. In Genome Research. And again, you see there's a general trend that as recombination goes up, pi goes up. Each individual circle here is a stretch of sequence in the human genome. So there you go. Well, this is true in humans, but is it just humans? No! Here it is in Drosophila. This was actually a classic study by Begun and Quadro in 1992 in Nature. This paper at the time of recording was already cited well over 550 times. It's a very popular study, but again, you see here on the x-axis is coefficient of exchange, which is essentially recombination. And on the y-axis here is pi, and again, you see this general positive trend. This, again, potentially suggests the spread of adaptive variants. We'll come back to this actually in the next video. Here's the same thing in various tomato species. That again, you see this higher pi in regions of high recombination. This wasn't as compelling, but it was weakly statistically significant, so that's cool. Yeast, in contrast, I don't actually show you a picture of it, but it actually doesn't really match the expectation. There was no significant positive or negative relationship. And it could be in this case that yeast tend to reproduce asexually very often rather than sexually, potentially reducing this opportunity. Now, let me ask, what does this overall pattern, not the yeast part, but for humans, tomatoes, Drosophila, what does this mean? Well, we see that there's a positive association between recombination rate and pi in many, many different species. Now, this relationship, again, is predicted from the action of natural selection, and not just any natural selection, but specifically the spread of adaptive variants, right? That we have this adaptive variant spreading, and we have a selective sweep associated with it and hitchhiking of nearby alleles. What this means, the fact that we see this positive association looking across the whole genome, is that selection's impact on genomes is huge. That basically selection is happening all the time. We have adaptive variants spreading within species all the time, and that's creating this overall pattern. Selection is strong. Darwin was right. Well, let me ask you, can we leverage this? Well, the answer is yes, that we can leverage this in an interesting way. Now, we expect the regions of low recombination to have low variation or low pi. Now, what would happen? This is actually a fictitious data point. I actually just drew it in here. What would happen if you see a very low pi in a very small region of high recombination? What does that actually mean? So let's say you have this, this region of very high recombination but has very low nucleotide diversity. What might you expect to happen there? Well, the answer could be, now there's a lot of possibilities. The answer could be, it could suggest there was a very recent selective sweep. It could suggest that basically very recently you had an adaptive variant within the last couple of hundred or couple of thousand years spread through all the populations of the species and eliminate variation at nearby neutral sites. What people do and have been doing and are continuing to do is we're using this to actually find genes that have experienced recent selection. Looking across the genome at regions that we know have high recombination and thus should have high pi, high nucleotide diversity. But when we see a small stretch of it that has unusually low pi, that could suggest that there has been a recent selective sweep and that there might be a very interesting gene to study there. Now, this overall pattern I've described to you in the context of this sort of adaptive variance spreading through the population, these frequent sweeps through the population. There is an alternative hypothesis. This alternative hypothesis for why pi and recombination rate may be correlated is referred to as background selection. We'll come back to background selection in the next video. Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about this association between recombination rate and pi overall. We suggested that one possible explanation for why there's this positive association between recombination rate and pi is because you have frequent selective sweeps across the genome. This reduces a lot of variation in regions of low recombination, making it so they have low pi, reduces very little variation in regions of high recombination, making it so they can sustain a high pi. Now, background selection is an alternative explanation for this pattern, and this is a continuing heated debate within the field of evolutionary biology in terms of its role relative to the other one. Now, I already gave you the positive view of the world, this idea that beneficial mutations around the genome cause this association between recombination rate and pi, this spread of adaptive variants. This was pioneered by these two fellows, David Begun and Chip Quadro. Chip Quadro is at Cornell, David Begun's now at the University of California at Davis. Now again, their idea is that there's very frequent selective sweeps on the genome, and that's what causes this overall positive association. Now the question is, what if beneficial mutations are actually very rare? Not just somewhat rare, but very rare. Well, if that's true, then you won't actually see a very strong overall trend, because you'll actually have a lot of low recombination regions that may still have very high pi, right? That if you're not having a selective sweep that's eliminating the neutral variation there, they might sustain a fairly high pi. Well, in response to this, an alternative hypothesis was posed by this fellow. This is Professor Brian Charlesworth at the University of Edinburgh in Scotland. He suggests that bad mutations are very common, which we know is true, of course. When a bad mutation happens, interestingly, what it'll do is it'll doom that allele and essentially the areas nearby it to eventual death. That over some period of time, even if you have a bad variant, it may be slow to get removed from a population, but it ultimately will be removed from the population. We saw this with some of those allele A1 simulations of detrimental variants that we did earlier. Now, importantly, the size of the doomed window varies with recombination rate. Okay? The size of this doomed window is going to vary with recombination rate. This process is referred to as background selection. Now, let me illustrate this for you. So let's imagine background selection in a region with low or no recombination. Now, what we have depicted here is background selection in a region of low or no recombination. So we have several neutral sites here. These blue ones are neutral. Okay, so this is essentially what diversity doesn't matter. And we have here a set of bad mutations that have arisen in this population. We see that we have a lot of bad mutations. We're assuming that these are fairly frequent. So what's going to happen? Well, all these chromosomes, if there's no recombination, all these ones that have a bad mutation are doomed, that they will not persist in the population. Eventually, they will go away. So the only ones that persist are these. These are the only ones that will contribute to long future generations. In that regard, you notice we've lost all variation at those blue sites. Now everybody's T and everybody's C because we got rid of so many different variants. This is what happens when you have a region of low or no recombination. What if you have high recombination? Now, in this case, I'm putting in something kind of like recombination hotspots here. That's what those vertical bars are. We have the same two variable sites that we had before with the same frequencies, and we have the same bad mutations. What happens now, we now just lose those segments that actually have the bad mutations. Now, these are not deletions. They're not like cut out of the chromosomes. It's just, and when you're looking at how much variation is lost from the population, those pieces would get lost. But the areas around it can persist because they can recombine away. So when you look at it now and you look at the two variable sites, you still retain quite a bit of variation. This is the important difference here, that a lot more variability is being retained because the bad mutations can be lost without dragging away so much of the rest of the chromosome. So what is the prediction of background selection? Well, background selection assumes, and it depends upon, having very high rates of bad mutation. We know that's true. What it predicts is that regions of high recombination, where you have hotspots that are close together or numerous hotspots, will preserve more variation, high pi. So we expect high recombination to have high pi, or low recombination to have low pi. That's exactly the same prediction as what we saw with selective sweeps and hitchhiking. So where are we? Well, this is kind of a big debate. We see this association between recombination rate and nucleotide variation as measured as pi. Now, it could be that's resulting from selective sweeps, that these new advantageous mutations are arising fairly frequently, and as they spread and fix, they're taking away most of this variation. And neutral theory says that this should be very rare, that the neutral theory does not deny that selective sweeps occur, but it suggests that they should be very rare. In contrast, background selection suggests that there's bad mutations happening all the time, and getting rid of them is what drags away variation. Now, the neutral theory has no problem with this, and this also generates the same prediction. We don't actually know which of these is true. This comes back to that neutralist selectionist debate that I suggest. This is essentially the new iteration of that. And this is very much an area of active research today. There's definitely continued disagreement. Here's a picture from a 2007 study looking at Drosophila simulans. The green there depicts levels of recombination. The blue depicts levels of nucleotide diversity measured as pi. And again, you see a fairly high correlation that when nucleotide variation is high, when recombination rate is high, nucleotide variation is high. Now, Begun argues that's mostly from hitchhiking, and he's shown that many beneficial mutations have actually been identified. Charlesworth argues that we know of many more cases of bad mutation. So let me give you a couple of perspectives on this. I actually contacted these people to get their input directly. Now, the recommended textbook that we have for you, the Freeman and Heron book, says the current consensus is that hitchhiking is probably responsible for the most dramatic instances of reduced polymorphism in linked regions, while background selection causes the less extreme cases. This is kind of a balanced view. Now, Begun tends to think background selection is not very important at all. He says there's no convincing evidence that background selection is a significant factor in Drosophila genome evolution at all. This is from an email dated 28th of February. So that's one end of the spectrum. At the other end, Dr. Charlesworth was a little bit less diplomatic in his wording, but he refers to a particular paper saying that they make a reasonable case that the reduced variability on the Y chromosome and 4th chromosome, these are chromosomes that have no recombination within species, can be explained very thoroughly by background selection. Well, this is very much an area that's still unsettled. Here's somebody else who's kind of in the middle. Suggests this controversy surrounding the relative significance of strongly beneficial and deleterious mutations in evolution has stimulated research activities for almost a decade. Despite efforts from many theorists and empiricists, fundamental questions are still open. Basically, the answer is, we don't really know. But again, still very much an active area of research today. How much of this correlation between recombination rate and nucleotide diversity comes from adaptive variation spreading or from bad mutations being eliminated from the population? Well, you can think about that yourself. Thanks for your time. I hope you enjoyed this video. Hello, and welcome back to Introduction to Genetics and Evolution. Now, we've talked about various evolutionary forces so far, and particularly emphasized two of them, natural selection and genetic drift. Now, a common question for people who are studying patterns of molecular evolution is, do particular genes evolve primarily as a result of the action of natural selection or genetic drift? And how many of the genetic differences that we see between species were driven by selection drift? Well, there are several challenges in trying to search for the signatures of natural selection at individual genes and infer the historical processes that operated on them. In this set of videos, we'll be looking at this particularly in the context of the gene changes that make us human. So what we'll do is we'll compare human sequences to those of our closest living relatives, which are chimpanzees, depicted here. Now, nucleotide variation exists within species and between species. In this illustration, we have a stretch of sequence. It's about maybe 15 bases long or so. We have 3 variable sites. This first variable site shows a fixed difference between species, meaning all individuals of species 1 differ at that nucleotide site from all individuals in species 2. So all of species 1 have a C, all of species 2 have a G. The second one shows variation within species 1, where some individuals are G and some are A, whereas species 2 doesn't have any variation. And at nucleotide site 3, we have exactly the opposite. There's variation within species 2. There's one individual that has a G, whereas all individuals in species 1 are the same. So we see this variation. How do we interpret it? Well, the big question is how much of the variation that you see within species or between species is neutral and primarily evolving by this process of genetic drift, and how much of it is selected, or can we identify any that seem to have experienced some sort of natural selection? Well, there's 2 schools of thought, as I mentioned before. There's sort of the neutralist school of thought, that most of the variation that you see within and between species is neutral and is evolving by the action of genetic drift. So if a mutation arises, its probability of eventual fixation is equal to its allele frequency. It bebops around and maybe gets lost, maybe gets fixed. In contrast, there's the selectionist that suggests that very little of the nucleotide variation that we see within or between species is neutral, that actually most of it is selected. So the big question for us then is what makes humans, or you could look at any focal species, special? Well, if we look historically, the human genome sequence was first published in 2003. It was available a few years before that. The chimpanzee genome sequence was published in 2005. And if you look at these genome sequences, they're very similar. On average, 98.7% of the nucleotides match up. Now, what evolutionary biologists want to do is to look at specific genes and say, for this particular gene, it seems like it was primarily evolving by genetic drift, and therefore those changes are, in terms of fitness, irrelevant, and others that seem to evolve primarily by natural selection, and thus may be important. It may actually, those ones that are important may be contributing to what makes us particularly unique, and different from our common ancestor with chimpanzees. Now, 98.7% nucleotide sounds like we're very similar. However, given our genome size is about 3.1 billion bases, we're actually different about 47 million nucleotide sites. So that's quite a few. Now, let me introduce to you the neutral theory of molecular evolution. This was popularized through the writings of Motu Kimura. Now, he suggested that most mutations that you actually observe, most mutations that get abundant and that eventually get fixed within a species, have no effect on fitness. So essentially, they're arising by mutation, and then they're bebopping around just randomly due to these stochastic forces, and they're spreading by genetic drift, or maybe immediately getting lost, potentially by genetic drift. They are not spreading by natural selection. Now, he doesn't argue against bad mutations. He is perfectly comfortable with the idea that some mutations are bad and immediately get eliminated, but that most of the variation that you see tends to not have any particular effect on fitness. There's a corollary from this, that if this is what's happening within species, then we expect most of the nucleotide differences between species, the ones that differentiate species 1 from species 2, presumably also spread by drift rather than spreading by selection. Now this, for many decades, has served as a null hypothesis, what we're essentially testing when we're looking at genome sequences or individual gene sequences. Now what the theory doesn't do is it doesn't say that all mutations are neutral. It very much acknowledges that there certainly are adaptive mutations. No neutralist would ever argue that camel's hump is neutral or anything like that, and obviously there's a genetic basis for it. It also, as I mentioned, it acknowledges common bad mutations. But again, that variation that you see, that variation that becomes abundant or eventually fixed, tends to have little or no effect on fitness. So there's variation that you see within or between species. Now some people really don't buy this. There was a paper published in 2008 by Matt Hahn, and he suggested the patterns apparent from multiple species at multiple loci make the case for rampant non-neutrality. So he suggested, and many others agree, that in fact there's very little of the genome that's evolving purely by the action of genetic drift. However, many, but not all, human pseudogenes appear to evolve neutrally. Pseudogenes are these things that used to be historically some sort of gene, but they essentially are no longer functional. They're just relics that are found in our gene. Now, the big question again for us is, how do we tell which of these 47 million bases that differ between humans and chimps evolved this difference by drift or evolved it by selection? Are they important or are they random? Well, there's a simple thing you could try to do, though it won't work, and that's just basically look at those areas that are most different. Basically pick those genes that are most different between humans and chimps and say, well, those are probably the ones that were important. So you could do something like this instead of looking at genes, looking at 1 million bases at a time. There are some stretches between humans and chimps that differ by more than 2% and others that differ by less than 1%. The problem is just because there's more of a difference doesn't mean selection's involved. And you can see there's a full distribution here. So some of these might have very large differences purely by chance. Some may have very small differences purely by chance. Not all base differences are important, and we can't just count them all exactly the same. Okay? Just to illustrate with you what I showed a second ago here, this is sequence differences looking along a chromosome. So this is the human-chimp divergence or difference in sequence. And you see there's a couple of peaks here that are potentially interesting, but there's a lot of valleys as well. There is an interesting correlation of this with recombination rate that we talked about elsewhere. But again, not all sequences matter. As I mentioned, base changes in pseudogenes potentially don't matter because they're not coding any protein. I also introduced introns back in one of the first genetics videos. These do not contribute to the final amino acids. Even though the sequence is found within the gene, they don't contribute to the final amino acid sequence and therefore don't affect the protein. So intron sequences are often thought to have no effect on phenotype. Not always, but often. Another set of sequences that may not matter so much are these codon third position changes, or specifically any change that's referred to as synonymous. Basically, there are multiple codon sequences, multiple triplets in the DNA and then eventually in the RNA that will make exactly the same amino acid. So looking at this, AGA encodes the amino acid serine. AGC encodes the amino acid serine. AGG encodes serine, etc. So we see this, even though we're changing that third nucleotide in this stretch here, it doesn't actually change the amino acid. And that's frequently true for these codon third positions. In contrast, the second position always changes the amino acid. So you see this triplet code AGA. So let's change that from AGA, which was serine, to AAA, that's phenylalanine, ACA, which is cysteine, ATA is tyrosine. So those are always non-synonymous. If you're changing the amino acid, you're necessarily changing something about the protein, right? So there is a high probability that a lot of these non-synonymous changes can affect fitness. In contrast, if you're not changing the amino acid, then it's more likely, though not certain, that you're not actually affecting the final protein in any detectable way, and these may be closer to neutral. Now, non-synonymous differences, as I said, affect the protein, and these non-synonymous differences in coding DNA will make a different amino acid stuck into the protein, and they are likely to affect the phenotype. Now, just because it's having an effect doesn't tell you the direction of the effect. These non-synonymous differences may be detrimental, right? They may be bad if the original protein was better. So you have a mutation from AGA to ATA that may make the protein in some way unstable and make it so you're not producing something you were supposed to be producing well. They may be neutral. Even though it is changing the amino acid, it may not actually affect the protein's function or structure in a noticeable way, aside from the fact that it is just a different amino acid in there. Or it may even be advantageous. Maybe this new protein, maybe it's better to have AAA than AGA, the amino acid associated with it. So these non-synonymous differences can be detrimental, neutral, or advantageous. In contrast, synonymous differences are presumed in general to be neutral or close to neutral. They're not affecting the phenotype. Now, what we want to do as evolutionary geneticists is to bin these changes. We want to, we have our three bins here: positive selection, negative selection, and neutral. So some of these gene changes that don't matter, they're neutral, we'll toss them into this bin. Many of the gene changes are going to be bad, so selection will prevent them from ever becoming abundant in the population or going to fixation, going to 100%. So they'll go into what's referred to as the negative selection bin, or purifying selection. Basically, this is the full set of mutations that are bad, that are quickly held at low frequency in the population and ultimately eliminated. So that's the negative selection bin. And then we have the occasional gene changes that are good. All right, these are ones that will be captured by natural selection and potentially spread all the way to fixation, to 100%. So these are positively selected. So we want to bin each of these in there. We want to know how much there is within and between species. Well, let me ask you a question first. Among these different things, if all base changes were neutral, would we expect there to be more synonymous differences between species or non-synonymous differences? If all base changes were neutral, if it didn't matter if you were AAA or ACA or AAT, which do you think would be more abundant? Well, the answer there may have surprised you. That in fact, there are actually more potential non-synonymous changes than there are synonymous changes between species, if we assumed all base changes were neutral. Well, let's explore this for a minute. Now first, let's think about what's happening here. When we're looking at differences between species, then a mutation has to have arisen within an ancestor and has to have spread, and that's what's creating this difference between species. So if species 1 has A at a particular site, species 2 has T at a particular site. Let's assume the ancestor had the A. What had to happen is, in this lineage leading to the ones that have a T, there had to be a mutation from A to T, and then the T variant had to spread. Right? So the mutation has to occur, and the new mutant allele has to fix or go to 100%. Now, the probability of fixation, if it's neutral, is 1 over 2n. It is the allele frequency of the new mutation. The probability of fixation, if it's bad, is much less than that. Okay? Now, interestingly, if you think about it, most coding mutations are going to be non-synonymous. There are many more mutations that will happen that will be non-synonymous than synonymous. The reason being, there's a lot more ways to be non-synonymous, because all second position changes are non-synonymous. Most first position changes are non-synonymous, and some third position changes are non-synonymous. Let's look at this here by looking at a codon table. So let's look at this codon table, and let's use this top corner one, UUU, just as an example. So this is phenylalanine. what base changes will change this phenylalanine, this UUU, into another phenylalanine? Well, in fact, the only change you can have is a U to C. If you change this last U to an A, it's a leucine. If you change this last U to a G, it's a leucine. If you change the first U to anything, to a C, to an A, or a G, it becomes a leucine, isoleucine, methionine, or valine. If you change that second base, the second U there, to anything, it becomes a serine, or tyrosine, or stop codon, or cysteine. So really, there's very few ways to change things synonymously. But there's a lot of ways to change things non-synonymously. That was a kind of an extreme example, but that's generally true looking across all these different amino acid codons. Still, even though that's true, synonymous sites usually accumulate differences faster. That when you look between species, typically speaking, you see more synonymous changes than non-synonymous changes, even though from mutation alone, there should have been more non-synonymous. What does that tell you? Well, that tells you that probably a lot of those non-synonymous mutations are bad, and they don't actually get the opportunity to spread. In contrast, the synonymous changes do have this opportunity. So here's a figure from one of your textbooks. This is showing differences between flu viruses over a 20-year period. Here's the accumulation of synonymous mutations. Here's the accumulation of non-synonymous mutations. So even though you're getting more of these non-synonymous mutations, they are selected against right away and never allowed to spread. So synonymous mutations happen less often, but are more likely to spread because there's not selection acting against them. So again, coming back to this question of how do we look at these differences between humans and chimps or any two species? Well, one is let's just look at genes that have many non-synonymous changes. Those are probably ones that matter, right? Well, not so simple. Unfortunately, mutation rates are not the same in all different genes. That some genes will get more mutations than others because of selection. And if you get more mutations, you're likely to accumulate more differences, whether they're synonymous or non-synonymous between species. So again, just looking at non-synonymous changes doesn't give you a lot of information about selection or importance. That's a decent candidate by looking at those, but by itself, you can't tell. The answer is, so we use these synonymous differences to scale the number of non-synonymous differences. Basically, synonymous differences should accumulate neutrally, and we can use them to scale for potential mutation rate differences between different parts of the genome. So what we can do is do some sort of ratio of non-synonymous differences to synonymous differences. And that is essentially estimating the non-neutral changes relative to neutral changes. From that, we can potentially interpret, though not completely cleanly, but we can potentially interpret the action of natural selection. So in the subsequent videos, we'll be doing this using two tests. One called dn over ds, the other called the McDonald-Kreitman test. I hope you'll join us. Thank you.